process of muscle contraction and relaxation has four major phases. The first phase is excitation. This is the process in which the nerve action potential is transferred to the muscle action potential. This is basically the animation that you just watched at the end of the last section. Next, we have to couple the excitation with contraction of the muscle. This is all the events that link the action potentials on the sarcolemma, or the muscle cell membrane, to activation of the myofilaments, actin and myosin, within the muscle cells, thereby preparing them to contract. Contraction is the step in which the muscle fiber actually develops tension and may shorten. And relaxation is after the work is all done and the muscle fiber relaxes and returns to its resting length. So let's begin by looking at the excitation of a muscle fiber. This is the process where action potentials in the nerve fiber lead to action potentials in the muscle fiber. You'll find this figure in your text. Let's break it down piece by piece. First of all, the nerve signal is going to open the voltage-gated calcium channels in the synaptic knob. Calcium will enter the synaptic knob. Calcium will stimulate excitosis of the synaptic vesicles, and acetylcholine, these little green dots, will be released into the synaptic cleft. Taking a closer look, on the other side of the synaptic cleft, we have these little purple guys, acetylcholine receptors. Here, the acetylcholine receptors will bind acetylcholine. Two molecules of acetylcholine bind to each receptor protein. This opens the sodium and potassium channels. Sodium will enter the cell very rapidly, which changes the resting membrane potential of the cell. At this point, don't worry about how resting membrane potential is shifted. We'll investigate that further when we study neurobiology later in the semester. However, we're basically transferring the electrical charge that came down the neuron into the cell by moving this ion, sodium, which has a positive charge, into the cell. This changes the electrical potential in the cell and causes an end plate potential. Again, you'll understand this more when we look at the neurobiology. But the basic story here is acetylcholine binds to the receptor, sodium rushes in, and that changes the electrical potential of the cell on the other side of the synapse, which causes it to propagate a signal. So the end plate potential then causes nearby voltage-gated channels to open, and this spreads the signal across the muscle surface. So the signal's now been propagated from the synapse here along the membrane to different regions of the muscle. This will continue as the signal spreads across the muscle membrane. So let's review with an animation from APR.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
Okay, so now we've got the excitation phase covered. The signal has been passed from the neuron to the muscle cell membrane. Now what? Excitation-contraction coupling refers to the events that link the action potentials of the sarcolemma, or the muscle cell membrane, to activation of the myofilaments, the actin and myosin. This prepares them to contract. This figure is in your textbook and will really help you understand the process. The action potential will spread down the T tubules. Remember, the T tubules were invaginations of the cell membrane that literally cause pipelines all the way through the cell that are continuous with the outer cell membrane of the cell. It allows the action potential to be spread all throughout the cell, very much like the wiring system in your house. The wave of action potentials spreads from the end plate in all directions, like ripples in a pond. When these waves reach the T tubules, they continue down them towards the sarcoplasm. Remember that the T tubules were coupled in a triad with two terminal cisternae of the sarcoplasmic reticulum. The action potentials regulate ion-gated channels in the T tubules. These are physically linked to the calcium channels in the terminal cisternae of the sarcoplasmic reticulum. The sarcoplasmic reticulum is literally packed with calcium, and so as the calcium channels open, they release calcium out into the cytosol of the cell. Once the calcium's out in the cytosol of the cell, it's going to bind to troponin. Now remember, we have these actin beads on a string, and they each have an active site on them. Tropomyosin was a fiber that blocks the active site only when troponin, this clip, was there to hold it in place. Well, calcium binds to troponin, and sort of like unlocking the clip, it releases the tropomyosin from the binding sites. Then the binding sites, here, these little divots, become available for the myosin heads. So let's review again. We've had the action potential being passed from the neuron into the synaptic cleft. From the synaptic cleft, it travels all along the membrane. Once it reaches T tubules, it spreads through these wires all throughout the cell, causing the sarcoplasmic reticulum to release calcium ions. These calcium ions will enter the cytosol and float around until they run into some actin and myosin. You can see the actin and myosin here in the myofilaments. Here, in a closer look, we see that calcium binds to troponin, which releases the clip so that tropomyosin can move aside and expose the active sites on the actin molecule. Once the actin sites are exposed, they're available for the myosin heads to clip into and row along the strand. So now let's put some motion on that. Here's an animation of excitation-contraction coupling.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
So now, take a moment to diagram the excitation-contraction coupling. The components you'll want are some sarcoplasm, T-tubules, sarcoplasmic reticulum, and then a small illustration of actin and myosin and the removal of tropomyosin by calcium's action on troponin. See if you can put those pieces together, and then we'll move on to the contraction phase. Now we're on to the contraction phase. Contraction is the step in which the muscle fiber develops tension and may shorten or contract. The way that muscles contract is described by the sliding filament theory. It says that myofilaments actually don't become shorter during contraction, but rather the thin filaments slide over the thick ones and pull the Z discs closer to each other. So in the previous phase, we uncovered the active sites on the actin molecule. This allows the myosin head space to bind. Now the myosin head has ATP attached to it. As this ATP hydrolyzes, it activates the head by cocking it into an extended position. So it moves it from this position to this position. This is the place that we use ATP. The ADP and the inorganic phosphate that results from hydrolysis of ATP remain attached to the myosin head. At this point, the myosin head is able to bind to the active site. It's been moved into the right position because we burned one ATP to cock it back. After the myosin head has bound to the active site, we see an actin-myosin cross-bridge. At this point, the myosin head releases the ADP and the inorganic phosphate and flexes, which pulls the thin filament past the thick filament. This is called the power stroke. Upon binding more ATP, the myosin is able to release from the actin, and the process is repeated. So it's the attachment of ATP to the myosin head that allows it to release. Each head performs 5 power strokes per second and utilizes 1 molecule of ATP per stroke. So let's review that quickly. We have the myosin head that's already been activated by ATP. Once the ATP is hydrolyzed or broken down, it allows the myosin head to cock back and attach to the active site on actin. After the cross bridge has been formed, the myosin literally rows, cocks the head back, and pushes the actin past itself. Binding of ATP to the myosin head again is what allows it to break the cross bridge and release from the actin. This process repeats over and over, allowing the Z discs to be drawn closer and closer to each other and thus shorten the muscle. Now let's put some animation on that phase and see if it helps clear up how this contraction phase works.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
So a picture may be worth 1,000 words, but a moving one, that's worth way more. Hopefully that clarifies what's going on. Now, you may ask, if all these myosin heads are releasing, doesn't it just slip and release? You have to remember there are hundreds of thousands of myosin heads connecting, all at slightly different times. So it's like pulling up a boat anchor. You go hand over hand over hand. In this fashion, some of the myosin heads are always connected to the actin. So now, using the figure in your textbook as a guide, diagram the sliding filament mechanism of contraction, including where ATP is utilized, how calcium plays a role. Now you can see, and maybe postulate what happens if there's a shortage of calcium. What if there's not enough calcium in our blood? What if there's not enough ATP? How do you think this affects contraction? Now we're into the fourth phase of muscle contraction. It's relaxation. Relaxation occurs when the work is done. A muscle fiber relaxes and returns to its resting length. Nerve signals that stimulate the acetylcholine release will stop. Acetylcholinesterase that we've visited before will break down acetylcholine. The fragments will be reabsorbed into the synaptic knob, as you've already seen. This means that the acetylcholine stimulation ceases. Calcium that was released from the sarcoplasmic reticulum is now being pumped back into the sarcoplasmic reticulum by active transport. Calcium binds to calsequestrin and is stored in the sarcoplasmic reticulum. Remember, it's ATP that was needed for muscle relaxation. It's hydrolyzed in order to get the contraction, but binding of an unhydrolyzed ATP molecule is critical in order to allow the myosin head to release from the actin. Once we remove calcium from the equation, it drops off of troponin, and that allows the tropomyosin to reblock the active sites. The muscle fiber is going to cease to produce any tension because there's no cross-bridge formation. Thus, it returns to its resting length. Part of this return is due to the recoil of the elastic components of contraction, as well as contraction of the antagonistic muscle. So your biceps muscle won't achieve full length until your triceps actually works to extend your arm. So let's come back to that question of what happens if there's not enough ATP. What's rigor mortis is a better question. When we have rigor mortis, it's hardening of the muscles and stiffening of the body. It begins about 3 or 4 hours after death. This is for a couple of different reasons. We have a deteriorating sarcoplasmic reticulum, and so calcium starts to leak out and enter the cytosol. What's the role of calcium? Well, calcium, remember, binds to the troponin so that tropomyosin can move aside. And thus, we've exposed the active site. If there's ATP still present, which there will be for a little while after death, then the actin-myosin cross bridges will form and the muscles contract. But as ATP comes into short supply, we find that the muscles can no longer relax. Remember, muscle relaxation requires the binding of ATP so that the myosin head can be released from the actin binding site. Thus, the fibers remain contracted until the myofilament proteins begin to decay. Rigor mortis peaks at about 12 hours after death and then diminishes over the next 48 to 60 hours, depending on the ambient temperature. This is one of the ways that investigators can tell how long someone has been dead. Pretty cool application of the details of muscle contraction, I think. Think through this one a little bit. Something else we might consider here are neuromuscular toxins. These are great topics for your end of semester presentation. We'll see some pesticides contain cholinesterase inhibitors. That means they bind to acetylcholinesterase and prevent it from degrading acetylcholine. So what does that cause? That basically causes paralysis, because if acetylcholine can't be degraded, it stays in the synaptic cleft and it continues to stimulate the muscles to contract. This is called spastic paralysis. Because of this, we could have possible suffocation. Think about what happens to the muscles of respiration. Clostridium tetani is a bacteria that causes tetanus. It's a form of spastic paralysis. Glycine in the spinal cord normally stops motor neurons from producing unwanted muscle contractions. The tetanus toxin blocks glycine release in the spinal cord and causes overstimulation and thus spastic paralysis of the muscles. Some plant poisons used by South American natives in their poison blow dart guns can cause a flaccid paralysis. This is a state in which muscles become limp and cannot contract. The toxin's called curare. It competes with acetylcholine for receptor sites. If the receptor sites aren't open for acetylcholine and they're bound with curare, which doesn't have the same effect as acetylcholine, the muscles cannot be stimulated. So when someone's been hit with a poison dart containing curare, they simply cannot move. And you're probably all familiar, at least with the name, Clostridium botulinum or botulism. We use this for Botox injections. Botulism is a type of food poisoning that's caused by the toxins secreted by this bacteria that we can often find in outdated canned food. It blocks the release of acetylcholine from the synaptic knob. If there's no acetylcholine, again, the muscles cannot contract and we have flaccid paralysis. This is why it's useful as a cosmetic. Botox injections are given to paralyze the muscles in the forehead, let's say. Now, do you think this is really a safe practice? I would question it. What if the Botox somehow diffuses into the bloodstream? Paralysis of respiratory muscles? Well, that really could be a problem. Anyway, think about some of these toxins. If you want to take one on and elaborate some more on it, bring us something exciting for a final presentation. These are great topics.